Hello everyone, welcome back to Dr. K. Primer lecture series presenting by Dr. K. Prem Sagar, that's me. Today we will talk about T7 DNA polymerase. T7 DNA polymerase and its features and how T7 DNA polymerase is modified to become a sequenase used in DNA sequencing. That's a chain termination sequencing method. So these things we will cover in this lecture. So you all know that E. coli DNA polymerase and TAC DNA polymerase are most studied. Out of which E. coli DNA polymerase, that's a polymerase 1 is the most studied and it is a prototype for all DNA polymerases. So here we will take the E. coli DNA polymerase domain organization to understand the domain organization of the T7 DNA polymerase. As you all aware that E. coli DNA polymerase is having 928 amino acid and its molecular weight is 109 kDa. And when you look at the E. coli DNA polymerase, it's having three distinct domains. The first domain is 5' to 3' exonuclease domain, and the second domain is 3' to 5' exonuclease domain, and third domain is 5' to 3' polymerase domain. So 5' to 3' exonuclease domain is present in the N terminus and the C terminus of the protein having the polymerase domain, that is 5' to 3' polymerase domain. In between them, it is having 3' to 5' exonuclease domain. So you all know that exonuclease domain, it removes 5' to 3' exonuclease domain, removes the primers in the replication, and 3' to 5' exonuclease domain useful in the proofreading of newly synthesized DNA molecule, and this 5' to 3' polymerase is involved in the elongation of the primer, that is nothing but DNA synthesis. So this all you know. By using this, we will learn about T7 DNA polymerase. That's T7 DNA polymerase is having 698 amino acid and its molecular weight is 84 kDa. And if you see, it is having only two domains when compared with the E. coli DNA polymerase 1. So that is, you see, this is having 3' to 5' exonuclease activity and 5' to 3' polymerase activity. So this enzyme is having only two activities, that is 3' to 5' exonuclease activity and 5' to 3' polymerase activity. It does not have 5' to 3' exonuclease activity. And you see N terminus is having 3' to 5' exonuclease activity. In the C terminus, it is having 5' to 3' polymerase activity. Here, in this polymerase domain, there is a small, there is a small binding domain that's called as TBS. TBS means what? Thioredoxin binding site. Thioredoxin binding site. So the T7 DNA polymerase is a two subunit enzyme. One is T7 DNA polymerase encoded by the T5 gene of T7 bacteriophage, that's here, and there is a small peptide, that's a thioredoxin, is encoded by the E. coli, whose molecular weight is 12 kD and 108 amino acid. So when T7 DNA polymerase binds with the thioredoxin, then it forms the polymerization. Otherwise, not at all. Right? So T7 DNA polymerase and thioredoxin, heterodimer club have a T7 DNA polymerase activity and other exonuclease activity. So the thioredoxin binds to the thioredoxin binding site in the thumb of DNA polymerase structure. You all know the DNA polymerase or any DNA polymerase have a right hand structure with the thumb, palm and fingers. Thumb is involved in the thumb always have a catalytic sites of the polymerase activity. This PAM is involved in the binding of primer to the primer and template, and these fingers are involved in the screening or scrutiny of incoming nucleotides. So this structure is very useful, and this is a structure is involved in the DNA synthesis, right hand structure. Always DNA polymerase have a right hand structure with the thumb, palm, and the fingers, right? Yeah, we'll see the same thing in the, and coming to the thioredoxin and the DNA polymerase, which is a two subunit complex, is performs the following activities. So the two subunit complex, that is T7DNA Polymerase and Thioredoxin, are necessary for polymerase activity, and double strand 3' to 5', exonuclease activity. So when a T7 DNA polymerase and thioredoxin form a two subunit complex, that complex is required having an activity of T7 DNA polymerase activity and 3' to 5' exonuclease activity, that is a double strand 3' to 5' exonuclease activity. And T7 DNA polymerase all alone, it can perform only single strand 3' to 5' exonuclease activity. 3' to 5' exonuclease activity. So here I am telling you that T7 DNA polymerase with the thioredoxin can catalyze the both polymerase activity and double strand 3' to 5' exonuclease activity. Two subunit complex performs the polymerase activity and double strand 3' to 5' exonuclease activity. And when it comes to the only single unit, that's a T7 DNA polymerase, can perform single strand 3' to 5' exonuclease activity. Here, two subunit complex, that is a T7 DNA polymerase and thioredoxin, have two activities, polymerase activity and double strand 3' to 5' exonuclease activity. And all alone, T7 DNA polymerase have a single strand 3' to 5' exonuclease activity. So in total, it is having three activities. One is polymerase activity, and second one is double strand 3' to 5' exonuclease activity, and single strand 3' to 5' exonuclease activity. The same thing we can see in the text. So T7 DNA polymerase is a two subunit protein consisting of polymerase from the T7 gene 5 from the T7 and 84 kDa, and a processivity factor from E. coli trxA gene from thioredoxin, that's a 12 kDa, and it is having three activities. One is a polymerase activity, and it processes strong 3' to 5' single strand exonuclease activity and double strand exonuclease activity, that's also in the 3' to 5' mode. Then here you should know one more thing, that thioredoxin do not have polymerase activity or exonuclease activity all alone. It cannot do anything. It's a only processivity factor for T7 DNA polymerase. Yeah. So the same thing I speak. You see that the T7 DNA polymerase active site is having two magnesium ions. As like any other polymerase, it is also requires the two magnesium ions for polymerization activity. And it's also have a right hand structure with the thumb, palm, and the fingers. The structure of the T7 DNA polymerase is also visualized as a right hand structure. Right? Now we will see the processivity. So the greatness of or special feature of the T7 DNA polymerase is it's having highest processivity. What is processivity? Processivity means ability of enzyme can add nucleotides, number of nucleotides it adds before dissociating from the primer and template complex. How many nucleotides it can add for a time, or how many nucleotides it can add for a one dissociation with the primer and template. That is called as a processivity. Some of the enzymes are having a less processivity. Some of them are enzymes having high processivity. So here the T7 DNA polymerase with the thioredoxin is having highest processivity. And without that, without the thioredoxin, it is having almost the processivity is very poor. That I want to tell you here. So you see T7DNA polymerase, and it is a primer which binds to the template. And you see this T7DNA polymerase is all in a single subunit, right? So it can add, if you provide the DNT base, it can add one or two nucleotides. That's it. Then it falls from the template and primer complex, right? So as a single subunit, it can polymerize, it can add one or two nucleotides, maximum of a dozen nucleotides. It can, then it will dissociate. See, single subunit enzyme synthesizes maximum of 1 to 50 nucleotides. Maximum is 50. When T7 DNA polymerase associated with the thioredoxin, then its processivity is increased enormously. You see, thioredoxin provide the DNT base. And still the enzyme is with the primer and template complex. So two subunit enzyme, that is a T7 DNA polymerase and thioredoxin, have a high processivity, means it can add minimum of 300 nucleotides to thousands of nucleotides for one association. So enzymes binds to the primer and template complex, adds the nucleotides, then dissociate. So the how many nucleotides are added for one association with the template and primer complex, that tells you the processivity or speed up the enzyme. So T7 DNA polymerase all alone, single subunit, have poor processivity. When it clubbed with the thioredoxin, it can add almost 300 to 1000 nucleotides per one association or one dissociation. Right? So this feature is very useful for a sequencing reaction. But the problem is, yeah, you see T7 DNA polymerase and thioredoxin binds to 1-to-1 complex with apparent dissociation consistency 5 nanomolar. You see, the binding of thioredoxin to T7 DNA polymerase increase the affinity of polymerase specifically to primer template. Because when a T7 DNA polymerase and the thioredoxin is as a association, as a two subunit complex, it will bind to the primer and template complex very strongly. Because it is binding to the primer and template complex, it can add nucleotides continuously. So processivity of two subunit T7 DNA polymerase is 100-fold higher than the single unit T7 DNA polymerase. So when T7 DNA polymerase binds with the thioredoxin, the processivity is 100-fold higher than the single subunit T7 DNA polymerase. So single subunit T7 DNA polymerase can add 1 to 50 nucleotides per one association or one dissociation, you say. But at the same time, T7 DNA polymerase and thioredoxin, that's a two subunit complex, can add 300 nucleotides to 1000 nucleotides per one dissociation with the template and a primer complex. You see, almost 1000-fold difference between them. Thioredoxin is increasing the affinity of complex to the primer and template complex. Because of that reason, the T7 DNA polymerase, two subunit DNA polymerase, T7 DNA polymerase having highest processivity. So you see, processivity describes the number of nucleotides continuously incorporated by DNA polymerase using the same primer and template without dissociation. right? So here the T7 DNA polymerase have high processivity. And one more reason is that when in the chain termination reaction, they use the dideoxynucleotides to stop or terminate the reaction. So T7 DNA polymerase do not discriminate between the dideoxynucleotides and deoxynucleotides. So these two features of the T7 DNA polymerase are ideal for to become a sequences. So one is high processivity, and the second thing is that it will not discriminate between the dideoxynucleotides as well as the DNTPs. It can add the same frequency dideoxynucleotides which are used in the chain termination reaction and DNTPs. So these two features are very ideal for sequences or sequencing reaction. But there is a problem with the uh, exonuclease activity. It is having 3' to 5' exonuclease activity. If that activity is inactivated in the T7 DNA polymerase, and this can become sequence is enzyme used in the DNA sequencing. High processivity of the T7 DNA polymerase with the thioredoxin, that is 300 nucleotides per association, 2000. And the second feature is ability to incorporate the modified nucleotides, which are important for DNA sequencing, on par with the regular DNTPs. It can add DNTPs or DDADNTPs or any other modified nucleotides the same rate. You need not to provide the high concentration of modified or less concentration of regular DNTPs. If provide the equimolar concentration, right, then it will not discriminate between the DDNTPs and DNTPs. So these two are ideal features for enzymes to become a sequences. But there is a problem is a 3' to 5' exonuclease activity. See, 3' to 5' exonuclease activity detrimental for DNA sequencing. Because why here, how it is a detrimental for DNA sequencing reaction is whenever reaction is exhausted with the DNTPs, then the 3' to 5' exonuclease activity and polymerase activity become net equal. So there is no polymerization. Instead of polymerization, it start degrading the growing chain. Right? The second activity is associated nuclease activity also cause the DNA polymerase to idle at a secondary structure. Whenever a single strand DNA forms the secondary structure, because of 3' to 5' exonuclease activity, polymerase activity also become idle at whenever it faces the secondary structure of DNA. And third problem is that exonuclease of enzyme can act on the labeled bands upon prolonged incubation, or it can cut down or it will break down the labeled nucleotides at ends because it is having a exonuclease activity. So these three problems are there with because of 3' to 5' exonuclease. If you can inactivate the 3' to 5' exonuclease activity, this T7 DNA polymerase is happily does the polymerase activity, which is in the sequencing reaction. So there are some strategies were used by the scientists to inactivate the 3' to 5' exonuclease activity. They use it to two strategies. One is a chemical strategy, and the second one is a biological mutagenesis. Here you can see. First, I will explain the problems. So this is a T7 DNA polymerase having a 3' to 5' exonuclease activity and polymerase activity and thyrodoxine. And you see, this because of this exonuclease activity can cleave or remove the labeled nucleotides from the ends or terminus. You see, it can remove. And the second problem is, as I told you, that whenever a single strand DNA forms a secondary structure like hairpin loops, and because of exonuclease activity, the polymerase become idle. Polymerase become idle. It will not involve in any more further polymerase activity. And third one is, you see, if you have surplus DNTPs, then it will allow the polymerase activity. It allows the polymerase activity. But whenever a reaction mixture, whenever a reaction mixture is having less concentration of DNTPs, then there is no polymerase activity. Instead of that, exonuclease start degrading the growing chain, or net synthesis is zero. So these are the problems with the exonuclease activity. So if you can remove this exonuclease activity from the T7DNA polymerase, and that can become a sequenase. So scientists have used the two different strategies to inactivate the 3' to 5' exonuclease activity. The first one is chemical modification, and the second one is mutagenesis. So when T7DNA polymerase is incubated with the iron, oxygen, and reducing agent, that's a oxidizing environment. In that condition, iron can bind with the exonuclease activity domain, and modifies the amino acids in the catalytic region, and once it is modified, then the activity is inactivated. Because the iron is able to bind with the metal ion, other metal ion binding site, right? You see, iron is uh, bound to the exonuclease activity site, then it is inactivating, it is modifying the amino acids around that, and inactivates the exonuclease activity. See, iron binds specifically to the exonuclease domain, produce reactive oxygen species that selectively modify amino acid residues necessary for the exonuclease activity. So this 3' to 5' exonuclease activity is inactivated. This is the first, sequencing one. That's a sequenase 1. So modified T7 DNA polymerase is a sequenase 1.0. The later on, but still, and when they're using this one, it is not possible to, in chemically, it is not possible to inactivate the every protein molecule, right? So if that is the case, some of the enzyme molecules may have the T7 exonuclease activity, that is again affecting the sequencing reactions. So that again they thought for a new completely inactivation means, so if you can remove some part of the exonuclease domain, then that may inactivate. So that gene may be used to produce the enzyme, and that enzyme is free of exonuclease activity. So then they tried the mutagenesis. Then this is what is the domain of exonuclease activity. This is the domain, and 28 nucleotides, 28 amino acids of the exonuclease domain is lost during the mutagenesis, which contributed the complete abolishment of exonuclease activity from T7 DNA polymerase. 28 amino acids were lost during the mutagenesis from this gene, which resulted in the loss of 3' to 5' exonuclease activity. So 3' to 5' exonuclease activity completely inactivated because of mutagenesis, where 28 amino acid encoding DNA is completely lost from its gene. So it is having partial exonuclease activity domain, which cannot perform the 3' to 5' exonuclease activity. So this version of T7 DNA polymerase is sequences 2. This was used for a couple of years during in a DNA sequencing reaction. But the disadvantage with this one is it is having you cannot do this chain sequencing reaction because it is not hot, thermostable. Later on, T7 TAC DNA polymerase, they have modified that. So the difference, TAC DNA polymerase is only drawback with the T7 DNA, TAC DNA polymerase is it can again discriminate between the DDNTPs and DNTPs. So there is a, when they studied the T7 DNA polymerase, there is a one amino acid which is not there compared with the E. coli DNA polymerase and TAC DNA polymerase, that is inactivated, and TAC DNA polymerase became a sequences 3 that is used in the Human Genome Project. So coming to the applications, T7 DNA polymerase, they are used in the preparation of radioactive probes. And the second application is, since it is having highest processivity, they are used in the synthesis of large fragments. So whenever you want to have a 10 kb or more, the T7 DNA polymerase with the Thioredoxin molecule used to synthesize it. So amplification of large fragments. And as usual, I told that you modify T7 DNA polymerase without 3' to 5' exonuclease activity used in the Sanger's DNA sequencing method for DNA sequencing. So this is all about T7 DNA polymerase features and the activities and modification of T7 DNA polymerase and application. So hope you like this lecture. If at all you like it, just subscribe for regular updates. If at all you have any questions or doubts, please let me know through the comment section. Okay, see you all with the one more video. Until then, bye bye.